Morphology and anatomy in relation to plant taxonomy. Morphology and anatomical studies have great importance for the identification and classification of the plant species. Here we are going to discuss about how in case of Ageratum conyzoides, the morphology and anatomy helps for the classification. Here, the aim of the experiment is to study the morphology and anatomy of the supply specimen, that is the Ageratum conyzoides. For this practical to perform in the laboratory, we need different materials such as needle plate, safranin, methyl green, microscopic slide, microscope, cover slip, pen, paper, plant specimen, etc. We will have to also prepare the fresh solutions of 70% and 95% ethanol for the anatomical, that is double staining processes. For the morphological study of the specimen, we have to observe the habit of the plant, that is where the plant grows and what is the habit, what is the habitat of the plants, and we have to note down the characteristic features of the stem and leaves of the plant, and also have to observe the reproductive structures, that is the flower of the plants, and have to collect these flowers and observe under simple microscope. For anatomical study, we have to make sections of the stem, transverse section of the stems, and these sections has to be make double stained so that the vascular bundles and other parts of the stem will be visible under the compound microscope. Sections of transverse sections of each specimen is taken separately and performed through double staining process and observed under compound microscope and noted their anatomical characteristics. For the double staining process, using forceps gently transfer the section into a small petri dish or well and immediate add a drop of safranin stain to cover the sample. Let the section sit in the strain for 3 minutes. Remove safranin by blotting it with filter paper and immediately added 70% ethanol. Let the section sit in 70% ethanol for 1 minute. Remove the ethanol was added more 70% ethanol for another 1 to 2 minutes to complete removal of excess amount of safranin from the section. After that, remove the remaining 70% ethanol by blotting and immediately add methyl green and let it sit for 1 minute. After that, remove methyl green by blotting. Immediately after this step, added 95% ethanol and allowed it to sit with gentle agitation for 1 minute. Removal of the ethanol was add 95% ethanol for another 1 to 2 minutes to removal excess of both the strains. Do not leave the section for too long in the ethanol as it is not remove too much of the strains. The section should appear translucent to blue. If it appears red, then the excess strain has not been effectively removed. After all these steps, gently transfer the section to a microscopic slide and with filter paper blot out excess ethanol.